So last time we talked about transpiration. You ran through the transpiration simulation and setup model. And so now we're going to actually examine how stomatal openings change, as well as some of the adaptations that plants have to alter their rates of transpiration. So first of all, what you can see here is a diagram of a stomata with the surrounding guard cells and the surrounding, further than that, epidermal cells. So there's the epidermis. This would probably be the lower epidermis of the plant, guard cells, stomatal pore, and then this is the closed version of the same thing. So you can see here in this diagram that when guard cells take in water, they'll swell up, their turgor pressure will increase, and they'll kind of bulge, and the stomatal opening will be pulled open. When they lose water, as in diagram B over here, well, their turgor pressure will decrease, and boop, the stomatal opening will close. This will happen as the plant starts to wilt. Let's say that it's getting too hot, too dry, losing water. It will do this as a preventative measure to stop further water loss. This is actually controlled by a plant hormone called abscisic acid. So when the dehydrated mesophyll cells sense that they are losing water, they'll begin releasing abscisic acid, or ABA, and ABA will cause efflux of potassium from the guard cells. That's just a fancy way of saying potassium will leave the guard cells. And just like if salt is being transported out, the water will follow by osmosis. Here, potassium ions get transported out, water follows by osmosis. So you can see that as the potassium leaves, that actually causes the water from the guard cells to leave. And this will happen in general too. As the plant starts losing water, its cells will lose water. Guard cells are cells, so they will, as they decrease in turgor pressure, start to close off those stomata. Light levels can also alter this process. So by actually causing potassium to now enter into the cells, we have potassium moving inward, because the potassium pumps can be activated by blue spectrum light. Potassium will send itself in, and then as that occurs, well, it won't send itself in. It will be actively transported in, and as that occurs, the water will again follow by osmosis. So make sure that when you're talking about movement of water that you're referring to osmosis as the process that describes it. It's usually worth a point to actually say osmosis, so make sure that you do that. So you can see here through these different mechanisms, the plants are able to change their opening of the stomata, so that way we can prevent water loss. We can make sure that stomata are nice and open during active phases of photosynthesis in the light. Some plants, many plants you may have found, are adapted to actually close their stomata around midday to prevent excess water loss in the heat of the midday sun in summertime. So at very bright light, this process actually gets reversed, and they'll start actually transporting out some of that potassium, so that way they don't have excess water loss. So if you did the transpiration simulation and found that some of your plants were actually slowing down transpiration in the light, that's not altogether surprising, because a lot of plants, especially those that live in warmer environments, have this adaptation in place. Speaking of plants living in warmer environments, slide my little picture thing down there. You should also know about some adaptations of plants in deserts as well as in saline soils for water conservation. So the objective is to be able to outline some of these. So these have two different names. Xerophytes are our desert plants, and halophyte, halo referring to salt, lives in our salty environments. And both of these are going to need to do certain things to prevent excess water loss through transpiration and other means. So xerophytes, first of all, maybe I'll go over here, live in our hot, dry environments like deserts. Many of these are cacti. Some might be other types of desert plants. And these plants have reduced their leaf area as the leaves get adapted to those rolled spines. So those little tiny spikes would be actually their leaves. They also many times have small hairs on or around those leaves to trap a layer of saturated air. Sometimes cacti will even look kind of fuzzy, like you want to pet them. Don't do that. Those are actually little spines underlying some of those hairs, but they help to trap some of that moisture. They'll have thick, waxy cuticles, especially when you think about like a cactus's little tiny needles. A lot of times they're like a brownish, almost plasticky looking substance on them. That's that thick wax surrounding these very thin rolled leaves. Many of them have low scrubby growth, although of course there's also picture here of a nice tall cactus, but a lot of cacti and desert plants tend to be short to prevent excess water loss because of excess surface area. They also have altered physiology called cam physiology. So what this means is that their photosynthesis is actually occurring at night when they can open their stomata more freely, so that way they're not going to lose as much of that water burning off in the heat of midday. And so this is just a little diagram here of a rolled cactus leaf. You can see that the waxy cuticle is indicated, but the stomata are sunken into these pits within this rolled leaf surface. So as the water exits out here, it gets trapped in this little rolled area and accumulates. So actually, even though the plant might be in a very dry desert environment, the environment right around those stomatal sunken in pits is going to be very humid. So that way you have nice slow transpiration so this plant can survive. Halophytes are plants that live in salty environments. Slide this over again. So in these saline environments, they're going to have difficulty with water uptake. If anything, they're going to tend to maybe even lose some water at their roots if they don't have special adaptations in place, because that salt is going to suck out the water. It's going to draw out the water by osmosis. So we want to stop that from happening. So some of the adaptations that halophytes have are similar. Some of them are different from what xerophytes do. So for one thing, they tend to reduce their leaves to scaly structures. Sometimes they're spines, but a lot of times they're more like these nice little flat scale-like structures. They also have special water storage structures located in the leaves, so that way they can store water there. If needed, if water starts becoming scarce and they can't support these extra vegetative structures, they'll actually shed their leaves. Sometimes they'll additionally shed them if they have a lot of salts that are becoming kind of toxic to the plant building up. As those toxic ions build up, they'll go through an abscission process and start shedding off their leaves. Much like the desert plants, they also have a thick cuticle. In this case, they'll have multi-layered epidermis, so that way, so that way it can help keep the water inside the plant as opposed to evaporating off. They also do something called cellular sequestration, so they can sequester or store salt in their cell walls or vacuoles, so that way it's not just circulating throughout the plant and messing up the plant's ability to transport water in and out of its cells. So they might have, like, let's say surrounding their stomata, this nice sunken outer chamber, and then an inner chamber that allows some water storage so some water can escape as needed. But it also, just like in the desert plants, has this tendency to pool up. That says sunken stomata there. So it has this tendency to pool, so that way the area right outside the stomatal opening is nice and humid and prevents some of that water loss. Things that you should certainly know are about the leaf structure, the thick cuticle, the multi-layered epidermis, sunken stomata. Another thing that these plants can do to help prevent some of the impacts of salt from their environment is that they can actually have mechanisms in place to exclude salts and other ions from coming in at their roots, and they can secrete salts from their stem tissue. So they can drop out their leaves if there's too much salt. They can secrete salt from their stems. They have several different adaptations in place to get rid of that excess salt if they need to. So these are different ways that they help conserve and keep the water balance appropriate within their little plant bodies. So take a look at these practice questions on transpiration. What is transpiration? Think about 2 environmental factors that will increase transpiration rate, be able to state those, and if you really want to take it a step further, be able to describe them. Think about 3 adaptations a xerophyte might have to reduce transpiration and explain how they achieve this. And then also think about the question, some plants close their stomata at night, and some also close their stomata around midday. So explain why this is advantageous to the plant. Just as a little bit of extra practice. Moving on to the next portion of transport in the xylem. So the first portion was mostly about transpiration. The next portion gets into how water is actually being moved throughout these plants. So first of all, this came up in the transpiration simulation. The cohesive property of water and the structures of those xylem vessels allows transport of water under tension. What this means is that as the water is transpired out, it creates a tension that draws the water upward. The adhesive property of water also generates these tension forces in the cell walls. So as, again, water is evaporating out, it gets pulled up along the side of those leaf walls. They kind of drag each other up like little like a little chain-like action up through those xylem vessels. In addition, this is also going to impact transport of water and mineral ions into the roots. So when mineral ions get actively transported into the roots, because the plant needs those things in order to survive, so there are specific active transport pumps in place, water will be absorbed by osmosis. Water is also being absorbed to continually replace that water that's lost through transpiration. So these different processes allow lots of water uptake at the roots. Some of the skills are that you should be able to draw the structure of primary xylem vessels in sections of stem based on microscope images. We're actually going to do that at another point when we look at microscope images of both xylem and phloem. And you should be able to discuss models of water transport in xylem using a simple apparatus as a model, like blotting or filter paper, porous pots, and capillary tubing. So we'll look at how these different things make good models of what's happening in those xylem vessels. So this is just not a cross section, but actually more of like a longitudinal kind of section through a xylem vessel, or really just a diagram. So what a xylem vessel is structured like, and I've used this analogy a couple times now, is like a drinking straw. Probably capillary tubing is a better analogy, but I think people are more familiar with drinking straws. So it's these nice rigid columns that are made nice and firm by a polymer called lignin. So lignin will form these nice ring-like structures to make a nice rigid tubing, so that way it can create a nice, smooth pathway for the water to travel up the plant. Water will move passively up the xylem by transpiration pull. As water is transpired outward, it creates this tension that causes the water to get pulled upwards under this tension process. And then again, the cohesive and adhesive properties of water help to facilitate this, help to kind of drag that water upward. So what we'll do is look at the xylem root observations through those models at some point, and you'll also work on the xylem and uptake at the root worksheets. So those are good things to do around now. The nature of science that we'll discuss more when we actually look at these models together are that we can use these models as representations of the real world. So the mechanisms involved in water transport can be investigated using simple apparatus that shows similarities in structure. So these porous pots allow that kind of nice wicking action that you would see going throughout the plant's leaves. The capillary tubings are a really nice model of the actual xylem vessels themselves. So this is actually very similar to the diagram that I showed on the previous slide with a little bit of discussion.
How do water and minerals get to the leaves? Although roots can exert a slight upward pressure, it is evaporation of water from leaves, a process called transpiration, that moves water molecules and ions up from the roots. Transpiration exerts a pull that is relayed downward along a string of water molecules from leaf to root.
So you can just see with that animation how this movement is occurring. So as water evaporates, transpiration pull is strong enough to draw the water upwards through the xylem vessels all the way from the roots to the leaves, allowing more water then to be drawn into the roots. Here's just a little quick version of that. So when the evaporation is occurring via the stomata, we're allowed to uptake the water and minerals at the root tissue. So minerals required by plants include magnesium for chlorophyll creation, nitrates for making amino acids, sodium, potassium, and phosphates. Plants will have specific active transport pumps in place in order to bring these into their roots. Many of these are going to come in through passive diffusion as well, but that requires a gradient. It's going to happen at slow rates because minerals are fairly low in soil and they get very quickly concentrated in those root tissues. So it's going to be slow, but some of it's going to happen through passive means. Some of it's going to happen through what's called indirect active transport, in which hydrogen pumps in root cells will actively expel those protons stored in their vacuoles. That will displace the positively charged mineral ions, and if you look up here, most of our mineral ions are positively charged. So when these proton pumps pass out these positively charged ions, it allows the displacement of other positively charged mineral ions, so they'll diffuse into the root along a gradient. We can also have anions like phosphates that bind to those protons that can be then reabsorbed. So this is sometimes referred to as mass flow. This is a little bit more of a technical process. The details of this are rarely asked in like excruciating, you have to be able to report upon all of this. What you should know is that proton pumps are allowing for this indirect kind of active transport. The specific mechanisms you don't have to get into too much, but just know that this facilitates movement of mostly positively charged mineral ions, but also absorption of some anions too. So this sort of shows this process happening here. So the hydrogen ions get actively transported out of the vacuoles of the root cells and into the soil. You can see if this is supposed to be our clay particle here, we can displace. So those little yellow dots are supposed to be the protons that are then displacing all of those mineral ions that were attached to it. And now they can be brought into the root tissue. So that way these are nice free mineral ions that we're looking to uptake. And as that happens, it will also allow the binding to some of those positively charged protons that are kind of free floating around. And that actually is it for today. If you look at the PowerPoint there, you might be able to see some hidden slides with some more information about movement into the roots. So if you're interested, you can check those out as well. And that's all I have for you.